So I have no problem with it. Imagine AI.
So, good afternoon, everyone. Today we continue with the CGTG seminars. Today we have Jana Obšteter, fellow Slovenian, who actually works at the Agricultural Institute of Slovenia, but is currently actually a visiting postdoc in our lab, so Highlander Lab, here at the Roslin Institute. She has used simulations on various species in the past, but in the past 2 years or so, she's been very much working on this insect breeding and simulator for honeybees. Apart from that, she works with ancestral recombination graphs, and today she'll be presenting about challenges and opportunities of insect breeding. So take it away.
Thank you, Jan. So hi to all, just an insurance policy. This presentation is more questions than answers, but this is just the stage that we are at in insect breeding at the moment. But we are sort of identifying the steps that we need to take to get to where other species are. So that's a step in the right direction. So all these images are AI generated, so I don't have a problem with authorship of them. Okay, so first I'd just like to thank the people that I work with the most. So they're mostly from the honeybee sector, as you might know. People here at the Roslin Institute, people back home in Slovenia, but also people from sort of New Zealand / Germany. Yeah. So I'll split it in 3 bits, and I'll start with the general challenges of insect breeding, sort of an overview of what's happening and actually what is going on. So insects are actually very important for ensuring global food and feed security. But there's actually multiple ways through which they contribute. So first one is pollination. Honeybees are one of the most important pollinators, but it's not just honeybees. You have bumblebees, all sorts of flies, solitary bees, wasps, so these wild populations that just live somewhere. Then we have insects for food and feed. And in Europe, these are the ones that's being cultivated, black soldier fly, which is the poster child, but there's also housefly, mealworm, cricket, migratory locusts. But then if we go to Africa, the number of species is actually much, much higher. And then we quite often forget these ones, but we also have insects for other products. So there are honeybees that produce wax and propolis, but also silk moths. So they're insects, and they actually producing silk is quite a big. Well, it's economically important because silk is quite expensive, and it's quite important in the eastern countries. They are also good for reducing environmental footprint, especially of food production. So we waste a lot of food. Around 30% of food is wasted from farm to fork, and insects can actually help with that. And then they also increase sustainability of modern farming systems because they have low environmental footprint in terms of how much water they actually use to grow them, and how much protein you get out. So that ratio is actually quite beneficial. So here I have a couple of pictures. The first one is sericulture, which means silkworms. The first picture is the silkworms that they only eat white mulberry. So that is the only thing they eat. So you have to have a lot of those trees. And then once they eat the leaves, you put them, they sort of cocoon themselves, and you put them on these spikes or any other medium. And you have to harvest them before they sort of chew their way out of the cocoon. So you have to kill them alive, because if they chew their way out, then the silk thread is split. And then those dead bugs can actually serve as food as well. And the second row is black soldier fly. The first picture is sort of how you made them in these mating boxes, and then they sort of grow in these boxes, eating basically whatever you give them. So what's the value of all this? So the value of pollination is 150 billion per year, which is 10% of total agriculture production, which is quite a lot. For insects as food, there's no such economical estimation, at least I don't know of it, but 2 billion people in 130 different countries eat insects on a regular basis as a part of their regular diet. Insects as feed is a growing industry that's expected to hit 2 billion per year by the end of the decade, and mostly that is aquaculture and pet food. However, although this insect farming is quite a thriving industry, insect breeding still has so many challenges. So honeybees are the most advanced. We have the most methods for them, but the breeding programs are actually very scarce. There's only one fully functional breeding program in Europe, in Germany, that includes 13 countries, but it's not as all-inclusive as it sounds, and that's only one. But then we have other insects where research is still focusing on basics, sort of how to rear them, how to best feed them. It's not that much about how to build a true breeding program, and there have been isolated impulses. So there's no like combined efforts to define breeding objectives, to have systematic data collection and stuff like that. So what do we need to do to change this? We've sort of identified the questions that we need to answer in order to get to a true breeding program. So first is, what is the population structure and diversity? So these populations were brought in from something that people caught in the wild. We have no idea what we brought in the labs. We are not sure whether this was picked from the nature, bred, or whether it's even fit for purpose. We just picked something that was willing to stay in our boxes, to be honest. And now we don't know how much of the, well, genetic diversity that represents. We have absolutely no estimates of global or even local effective population sizes. Some attempts have been done for honeybees, but the numbers are all over the place. And this is the problem, because we don't know whether we are in the 100,000s, whether we are in the 10,000s, whether are we in the thousands. We have absolutely no idea. So there's no information on the effect of this domestication, and I'm calling it domestication because none of these insects are truly domesticated animals. But all these information is needed if we want to start a sustainable breeding program, and if we want to also simulate them, because if we don't, then we can't represent their genome really well. Then the next problem is the breeding objective. So for the breeding objective, we need to understand the biology, how the species behaves. We need to understand the production system, and we need to understand the market needs and wishes. Currently, and I might be wrong, there's little communication between what industry needs, what research needs, what market needs. So industry knows about mass rearing, the problems it runs into when it's trying to go big. But then it doesn't know a lot about breeding. On the other hand, researchers who do it know about breeding, but have no knowledge about these problems that industry is running into. And then market needs are a third component that's not really well researched. So a consequence of this is that the breeding goals are not really defined. In the edible insect sector, it's in the space of weight and protein yield, or maybe you are selecting for fat content or something like that. In honeybees, it's a bit more defined. So usually we select for honey yield, swarming tendency, hygienic behavior, and aggressiveness. And although these seem quite well defined, the scoring of this is very primitive. It's 1 to 4, subjective. So everyone gives them 4 for calmness, because it's like, oh, I want my bees to appear nice and calm, so everyone will buy them. The hygienic behavior is not defined at all, because there's multiple stages that could affect it. So it could be which bee starts picking the infested cells first, which one carry them out, which one smell it. So this is different traits. It's not defined. So yeah, looks great. Isn't that great, really? Then the next problem is phenotyping and identification. So the first problem is that these animals are tiny, right? So if you try to tag them and measure them, you might run into problems. So in honeybees, we put these little tags on the queens, tiny little discs with tiny little numbers. So you have to be quite skillful to do that, and then you have like a bunch of tagged queens. If they lose it, well, then you don't know which queen it is. But insects can get much smaller than a honeybee queen. So you can have a problem with that. Then there's a short lifespan. So you need to measure and process the information before the little insect dies, which is, you could have very little time if you want to keep it for the next generation. Then the next problem is there's a social organization. So we have interactions between these animals that we don't know how they affect phenotype, but also that what we measure in these boxes might not reflect what they do when they go into industry conditions. So if we measure something in a box, it grows well, we put it on we scale it up, and it just doesn't perform well. So a more realistic alternative is a batch colony phenotyping. Sort of you phenotype a group. You measure, you weight a group of larvae. You don't measure each larvae, but you measure a group. But then the problem is how to model it, because it's going to be, first thing is there's a common environmental effect. There's for sure is G by E that we don't know basically nothing about in insects. There's also G by community, which is the social interactions within the group of insects that we also know nothing about. So individual phenotypes are not an option. So we're gonna go with batch, but then we need to figure out how to model them. And then the next problem, okay, you've selected the individuals, but then you need to mate them. You need to mate the little female and the little male. How do you do that? Well, you need to somehow control mating for assessing relatedness, but also ensuring genetic gain. But again, as I said, problems with identification. Who is that female that you've selected? And you could have multiple matings or polyandrous matings, which means one female mates with multiple males. And it usually happens somewhere in the air, and you have no control of that, right? If you just leave them to fly and mate in the air, then who mated? There are some solutions that have been developed for honeybees. They won't apply to all insects, but I'll just skim through them. First one is artificial insemination. Yes, we do have that in bees, but you have to be super skillful. This is getting a sperm from a drone, and there's very, very little people who can do that. You have to collect sperm from 10 to 15 drones, and then you have to inseminate the queen. So here you squeeze the drone, and basically its genitalia comes out. But when you inseminate the queen, they basically tie the queen down, right? Like spread her legs, this tiny queen, and then put the sperm into her. So not a lot of people, and this cannot be done routinely, right? Next one is geographic isolation, where you create a breeding station either on an island or an alpine valley. So somewhere where there's no other bees around. Usually they don't live in alpine valleys. So what happens here is you select something that's called a sire queen. So one single queen cannot produce enough drones to inseminate everyone. So you sort of produce more queens from her. So they are not clones, they are now recombined queens. So this is the best you can do. There's multiple individuals here. And then they are mated, and then they produce drones. But the fact that they are mated doesn't impact this, because drones are haploid and from unfertilized eggs. And then you get a bunch of drones coming from these sisters, and this is the best you can do to control the sire site. Then the third one is a temporal isolation, where you bring out the queens and the drones after all the other bees have gone to bed, which can be 4 or 5 in the afternoon. And they do this train of queens, which they bring them out of this train. And it's because they have to be at the same position in space every day, otherwise they get confused and they are not oriented in space. So they have them on a train, and then after all other bees go to their hives, they bring them out, and then they take them back in, and then they keep them in the dark until like 3 or 4 p.m. And then the last one is the biological isolation, which is you just overflow the space with your desired drones and hope that the probability works to your advantage. Okay, so this works for honeybees. Does it work for other insects? Yeah, we don't know. And all of these problems then affect the estimation of breeding values, because the first thing is we have this batch or colony phenotype. The next one is we don't know who the mates are. We have this community interactions. And there have been some solutions developed for honeybees, but it's only for a specific setting where they have this breeding station or AI mating. Otherwise, you can't really predict the breeding values accurately. Okay, so because we don't know what to do with this, we've established this cost action called InsectIm that kicks off in October. Its cost actions are pan-European actions for knowledge transfer. So we will aim to transfer knowledge from animal breeding to insect breeding and between research and industry. And if anyone's interested, either an animal breeder, aquaculture breeder, insect breeder, company, innovator, you can actually apply to our working groups on this link here. And this is done through eCost. Also, this has a website, so if you just type InsectIm cost, it throws you on a website, and then applications can be done through there. Okay, since I work on honeybees, I'll dive in honeybees a bit more, and some of you already know this. But I'll kick off with some fun facts. So honeybees don't see the color red because we see in a red, green, blue spectra. They see in green, blue, ultraviolet spectra. So they don't see the red that appears black to them, but they see the ultraviolet. But yet we end up with hives that look like this. They don't see the red, but we keep forcing the red on them, right? It doesn't work that great. The next thing is honeybees are actually evolutionary advanced, and they can actually do simple math. They've shown this a couple of years ago. They trained them in a labyrinth. They can do addition. But then we get guys like this one here, if you focus on the error. It didn't really get it. Maybe because it was red. So they don't always get it right. And then the last thing is that they are really hardworking. A single colony can produce up to 250 pounds of honey per season. So they eat most of it, right? So they need around 70 kilo per colony to survive, and then the rest is yours. But it's not true for all the bees. Drones actually do absolutely nothing. So males just sit around. They have to be fed. They can't eat themselves. They're just there for their reproduction. So honeybees, on top of all this stuff, have a social organization, which means that there is one queen that manages the colony through the pheromones, and she also reproduces. We can have up to 60,000 workers that do the tasks. Cleaners, guard, bring pollen, bring nectar. And in honeybees, the roles change through their lives. So the older they are, the most likely it is they're going to be outside the hive, because it also means that they're going to be most likely to die outside the hive, which keeps the hive clean. And then we can have up to, I wrote 5,000, but it's several thousand drones that do nothing, sit around and reproduce. And they're quite big. The drones are bigger and have these big, bulky eyes. So what are the implications for inbreeding of this social organization? So we have this colony phenotype. And we have the interaction between these bees, because they're social insects, but what kind of interaction there is, we don't know. And also the phenotype is influenced by the queen, which is a maternal effect, and worker effect, and they're usually negatively correlated, as it is common in other species. The next peculiarity is that they're haplodeplete. So females are diplete, drones are haploid, and that's why we call them flying gametes, because they're just gametes of the queen that are flying around. And the implication is that this relatedness mirrors the X chromosome inheritance, and that is why the full SIPs, so maternal and paternal, are not related 0.5, as you would expect, but are related 0.75. And within a hive, we call that super sisters. And the other implication is that the Mendelian sampling term is not independent anymore, but it's actually it is dependent on the father of the workers, because the father's genome doesn't recombine when they pass them to daughters. And because this is quantitative genetics, I've put in a couple of formulas. So what is the consequence of the social organization and haplodiploidy for the estimation of breeding values? And this method was developed by Braskamp and Baima. First one is that, as I already said, there are two genetic effects. This model looks quite normal, to be honest. There's a worker effect, which is a direct effect, and the queen effect, which is the maternal effect. The problem is that these two are always confounded. So in mammalian species, you would get one mom that would have multiple offspring with phenotypes. Here, one mom is always linked to one colony. So the maternal effect is always confounded with the direct effect. One queen cannot have multiple colonies with phenotype, and one worker group will have only one queen. So there's a problem how to parse these two apart, and you have to have really good data to parse them apart.
Jana?
Yeah.
How does this work if the Y is, you said, on the colony level?
Yeah.
So how does this link to individual worker effects?
No, this is the work group.
Okay.
Yeah,
Thank you.
I'll explain now. The second problem is we have a non-standard A matrix and an A inverse, so we can't use the standard software. You can't open R, load the package, and just run it. And it's because some individuals are groups. So worker queen is a single diploid organism. Workers is a group, so aggregate of individuals, and so are the sires. So as I've shown you before, sires are the group of multiple sister or unrelated queens. So the A matrix then cannot be just computed regularly. So if this is a breeding value of a queen, for example, half of it comes from the dam, half of it comes from the sires, but this is now a group, and then plus Mendelian sampling. The variance of this sires group, it's not variance 1 plus F, but it actually contains some other stuff in here. And here some assumptions need to be made, which is what is this S, which means how many queens have mated, how many sires are in that, how many queens are in the sire, basically, and what's the relatedness between them. So it could be unrelated if this is just somewhere in the air and it's random queens, but it could be very related if we have this group of sisters. And then the variance of the Mendelian sampling also has an additional term, so coming from the dam, coming from the sire, but this is which of those queens in the sire were sampled. So we have an additional term in the Mendelian sampling. And the second problem for the A and A inverse is, as I already mentioned, Mendelian sampling is not independent. So if we write this A matrix as a product of these three matrices, T is the one that links the individual to his ancestors, and D would contain the Mendelian samplings. Usually the D matrix would be the diagonal, but here it's a block diagonal because we have these groups of full sisters that have inherited non-recombined DNA from the dead. So these are the problems for the estimation. And right now there's only one sort of custom-wide software that runs this, but otherwise you're on your own. The next problem is, it's not over yet, they have this complementary sex determination, so they don't have a sex chromosome. They don't do it like mammals, but they have one single locus on chromosome 3, where females are diploid, males are haploid, and if the offspring is diploid and heterozygous, it's a working female. If it's a hemizygous, which means haplo, how do you call it? It's a hemizygous, has only one chromosome, then it's a regular working drone. But if we got a homozygous diploid, those are then diploid drones, which do live, but then the workers somehow smell it, and they kill them immediately because they know something's wrong. So they kill them as larvae. They're very efficient in that, and I don't think no one knows how they smell it, but they somehow know. So those guys die, and this is a consequence of inbreeding. So if you have a lot of inbreeding, this happens. This all should be full, but all these guys in between were actually killed because they're homozygous drones. So you get a lot of empty spaces in this, I don't know how it's called in English, in the hive where actually should be larvae, but it's now empty. So this is not going to be a strong family. You don't have a lot of offspring. And then you also have colony events. So if you're an individual, what can happen to you? You are born, then you mate, then you produce something and we measure it, and then you die. But if you're a group of individuals, a lot of things can happen to you. And they affect the diversity, but also gain. So here I have just two of them. So one is when bees swarm, you probably heard about this. This is their mean of reproduction. This is how they reproduce. When the hive is too crowded, and when the resources are abundant, so when they have a lot of food, they will reproduce through swarming, which means that half of the workers will leave with the old queen, and then this rest of the workers will raise a new virgin queen. And this is basically how they reproduce. From one, they go to two. And then there's superseding, which means old queen dies, the new virgin queen comes, is raised from one of the larvae. The problem is that once this happens, in the hive, you now have a mixture of workers from the previous queen and from the new queen. And now you measure phenotype. Who do you assign the phenotype to? You have two queens and a mixture of both offspring in the hive at one time. And if you measure at that time, it's who is it for? Who is it from? So this can be a problem. However, when they swarm, they don't produce honey for you to yield, so that's better. When they supersede, they still produce they work normally. So it's a question who the phenotype is assigned to at that point when the old queen dies. And why do we need breed honeybees? So recently there have been increased honeybee colony losses. First one is increased pathogen load. You might have heard of Varroa. So Varroa was associated with the Asian bee. But then, because we travel a lot, we brought it here, and our bee is not, the European bee is not resistant to it. The African bee knows how to deal with it, but ours doesn't. We have loads of viruses, bacteria that has been, and that all this is caused by this migratory beekeeping and taking bees from UK to New Zealand and from New Zealand to America and stuff like that. We have habitat loss and habitat fragmentation, and there's agricultural intensification, which comes with increased use of pesticides and herbicides. Just, I think 2 weeks ago, there was a story from Croatia where they lost like millions and millions of bees. And it's because they wanted to control for mosquitoes and they sprayed something, they killed all the bees in the area. So like lots of beekeepers just lost all of their colonies. But there's also climate change. So this year, for example, in Slovenia, it's been raining a lot. Almost every day there's no honey. There's nothing for bees to eat because all the pollen is washed away. Last year it was too hot, so they couldn't fly out because they would be burned to death. So this climate change really affects the insects quite a lot, and we can't put them like under in a shed or under a roof because we just can't. They're very dependent on the environment. Okay, and lastly, I'll talk a bit about our efforts to try to contribute to this insect breeding story. So we started off by saying, oh, we need to optimize the breeding program. How would you normally do that? Well, you'll simulate a couple of scenarios and see which one works best. Great, made a plan. Is there a simulator? There isn't a simulator. Okay, so we had to build one. So we built this SIMplyBee, which is a stochastic simulator of honeybee breeding programs. For those of you who know AlphaSimR, it's an AlphaSimR extension, and it accounts for all this honeybee weird stuff. So it simulates the honeybee genome according to the demography. It simulates their biology. So we simulate haplodiploidy, polyandry, caste system, CSD mortality. We simulate all these colony events. So they swarm, they split, they collapse. But also the quantitative genetics bit, so it is possible to simulate the direct, indirect genetic effects and also to get a colony phenotype out. So in AlphaSimR, you have the basic class is a population where each individual would have a mom and a dad, a genome, and a corresponding genetic and phenotypic value. So we did the same here. Each bee gets a genome, and each bee has a mom and a dad. Although in reality we might know it, in simulation we do, and each bee gets a phenotypic value. it might never be expressed, but it is there. But then because no one works with one single honeybee, because they live in colonies, we had to produce this colony class where all these are now, we have a queen, we have workers, we have drones, they behave a bit differently. They all have their own genome and have their own phenotypic value. And lots of things can happen to them. So if we swarm them, they don't produce honey anymore. If we split them, a part still produces honey. If they supersede, they produce honey, but the genetics in the hive is now mixed. And they also have a location. So each colony is somewhere in the space. And also because you usually don't work with a single colony, like you wouldn't work with a single cow, but you would have a herd of them. We have a multi-colony, which is just a lot of colonies in here. And this can represent like an apiary or an area or an age class. So what is the novelty of this simulator? That now we are allowed to model the social interactions, right? So bees somehow interact. They in reality, they don't have their own phenotypes, but what gets out is one single value. So in this software, you can combine them into a colony phenotype, but how you combine them is up to the user, because we don't know what the interaction between bees is. So it could be linear. And this has been shown for honey yield, that sort of it increases with the number of bees and it decreases quite linearly. So you could do something like colony pheno is just the sum of the queen effect plus some mean or sum of the workers. But it could be more complex, sort of a threshold model. And I'm calling it a threshold with the quotes because threshold model is something else. But sort of for aggressiveness, it's been shown that when a couple of bees that are more prone to it start stinging, then just all of them start stinging because there's something in the air. So you just need a couple of bees with a really strong phenotype or genetic value for it, and then the colony will express the behavior. However, is this true? We don't know, but as more research is done in this area, we are able to model it, but also we are now able to model it and then check how this sort of reflects what we see in real life. And people are starting to collect individual bee phenotypes, although that seems weird because how much honey does one bee bring? We can't know, but they are sort of doing individual phenotypes for drones, sperm quality and stuff like that. The other thing we've added is the spatial mating. So a bee here in Edinburgh will never mate with a bee in England because they can fly max 10 kilometers, but that's max if they're really pushed. Usually they would fly around 5 kilometers. So if they're close together, they're more likely to mate. And how it happens is all the drones within like 5 or 10 kilometers would fly to one cloud. And it's been hypothesized that they orient themselves based on the landscapes. Like if they see a very tall tree, they will gather around the tree. Or if they see some depression in the landscape, they will gather there. This is like still a theory, but they do orient themselves somehow. They also follow a drone, like a mechanical drone, if you just fly it, so I don't know what their clue is, but they somehow manage to organize themselves. And then the queens fly to there, and then everyone in this cloud sort of has the same probability of mating. So we do this in the simulation as well. Each colony has a location. So we say mate in the radius of 10 kilometers. And then we run this approximate nearest neighbor through this R package, ran, that uses a C++ package N. It's very quick. And this one just finds mates in the radius, I would say 5 kilometers, and just mates accordingly. So we get the spatial dependencies of mating and then the genetic and the phenotypic values. So what we can model now with this SIMplyBee is, for example, we can model the CSD variability or the inbreeding. So here we have we ran 10 years of two pure populations, so Carnica and Mellifera, two subspecies, sort of breeds of honeybees, and then one was crossed. And then we were checking how much homozygous brood we observe, so how much of the offspring is those homozygous diplets that die, how much of the CSD alleles do we see per colony and how much do we see per population. So now we can model something like this and see how these populations behave. Or we can model relationships within a colony. So this is sort of, we check the IBD expected, IBD realized, and IBS, and sort of we see 3 distinct peaks here in blue, which is worker to worker. 0.25 are the half sisters, sort of same mom, different dad. 0.75 are the full sisters, same dad, same mom. And then 0.5 would be those that have the same mom, but the brothers, the fathers are brothers. And these ones are sort of the full SIBs in the mammalian sense, which would be at 0.5, sharing 0.5% of their genome. Well, these are twice the relationship, the kinship values, I might say that. So half of this, that would be the full SIBs in the mammalian sense. But they are actually very rare in a colony because the drones have to be brothers, and the probability of that is not very high if they fly somewhere to the air. Okay, so this is basically insect breeding in short. Like I said, there's more questions than answers. Just to conclude, so insect farming, not to be confused with breeding, well, they go hand in hand, but farming is actually quite advanced. It's assuring food security, reducing environmental imprint, but the breeding aspect of it is actually still quite poorly understood, and the methods are not really developed. So the major issues is what is the genetic structure, sort of what we brought in from the wild, how do we tag them. Someone gave suggestions to print QR codes on them, I think, didn't they? But then I don't know how that worked. How do we control mating for any of these species? And this is where we sort of need the entomologists to tell us how they truly mate, because I was just talking to some entomologists and they said, oh yeah, the black soldier flies actually need the UV light to mate. Who knew? Then the non-invasive phenotyping, how do we phenotype them without harming them? Because if you want to phenotype, if you want to genotype a queen, if you harm it, they will kill it, because if the queen is imperfect, she's dead, she's gone. You can clip her wings, they will allow for that, but anything else, she will be dead. Then how to model phenotypes to estimate breeding values, but also how to define breeding objectives. So in this space, honeybees are most advanced, but it's not a lot of breeding programs regarding this method. Regardless of these methods, there's only basically one truly functioning one. So again, we've started this action if you want to join, and we also wrote this software as a first step of our contribution to building better insect breeding programs. So with that, I'd like to thank you, and if you have any questions about bugs, I'm happy to answer.
Any questions in the room? Go ahead.
I want to know if you have, there is a SNP chip available for bee genotherapy.
Sorry?
There's a
Is there any
SNP.
SNP chips available for
Yeah.
bee genotherapy?
Yeah, so there's 2. One has only 4K SNPs, only 4,000, and it's not really good. And there's another one, has around 100,000 SNPs, but it's still very expensive. So it's not like for cows where you can get a genome for $20. It's still expensive. However, honeybee genome is super small, so it's only 250 millions. So sequencing is not that expensive either. Well, compared to like genomes that are 3 billion base pairs, this one is like 10 times less. So yeah, it's not a lot of use of these chips.
Okay,
Yet.
and for the genotyping, is it individual genotyping or colony genotyping?
Depends. Usually you don't do individuals. So if you wanted to genotype the queen, you would take her drones, because the drones is just recombined queen's genome. There's nothing else came in. So basically, take the drones, blend them, basically. and you send that to pool genotyping, but that is actually the queen's genotype. They also do workers, sort of pool samples of workers, but in that you have the queen and the drones that she mated with. It is possible to genotype a queen through, like you clip her wings, or you can also take the feces, and there's another thing that it's called exuvia. It's another liquid that they sort of release that has her DNA, but it's not gonna be like, how's Matea calling it? It's not gonna be high quality DNA from those liquids and feces. So you would sort of take drones and do pooled genotyping.
Thank you.
Can you mention the low coverage sequences offer?
No problem.
Yeah, so they're doing, is it Brock that has this? So Brock Harper at Purdue has this project that does low coverage sequencing for I don't know how many million bees they intend to sequence, and it's basically low cover 1x, 2x, and then done by imputation. So that way you can get. However, the recombination rate is really high, really high, 10 times higher than in human. So I'm hoping the imputation works well.
The software that you developed, can it be applied to any other insects, or is it specific for honeybees?
It can be applied to social insects, which is ants, termites. You have some social mammals, no, not mammals, rodents as well, like mole rats. Perhaps works for that as well. We would have to tweak the simulation of the genome, but yeah. Ants would probably work.
All right. You said that bees are not truly domesticated, but what do you mean by that? Because for me, domestication in terms of plant breeding is developing traits or selecting for traits that sort of fit the human purpose. So in this case, bees have been bred to be more calm.
Yeah, but I think in the animal sense of domestication is associated with brain changes. Like dogs, cats, other domesticated animals, there have been brain changes associated with domestication, which means that the animal is now cannot survive without you. Bees can easily survive without us. They do live without us in nature. We have wild bees. It's not like you never find wild cows, right? Cattle, but you can find wild bees. They live in nature and they are perfectly able to survive on their own. That's why they're not truly domesticated.
Okay. There's a comment. Is there a comment?
Yeah, do you want to read?
Or maybe I just put it up. Yeah.
The swarming or the supersedure, if we would know at which point and how much honey is produced from each queen, which we don't, but if we would, we could address up to the Z matrix. So at the moment we allocate one phenotype to just one queen, a worker group. We could say 50-50, instead of having 0-1, we could have proportion to the Z matrix.
Yeah.
But of course,
If we knew, yeah.
Yeah.
I think that is why also, well, you don't get honey from swarmed bees, but you also wouldn't take it. I think it's that reason maybe.
Okay. Do you want to read it yourself?
Okay. Is the increasing domestication of honeybees a good idea? On one hand, production can be increased, welfare through disease resistance could be increased. On the other hand, their survival would then depend on more intensive interventions by humans. Yeah, I think there are two different aspects to look at bees. Are they sort of there for economic reasons, or are they there for preserving diversity? If you're a beekeeper, you probably want them to rely on you, but to give you more honey. If you're a bee conservationist, then you probably want the other thing. But it is one thing that the environment is changing too quickly for them to adapt so quickly, and we are bringing in a lot of pathogens. We're bringing in varroa. We're bringing in the Asian hornets, and those are not natural things. So the bees can't adapt that quickly. Like European bees have no way of dealing with Asian hornets. They have ways of dealing with European hornets, and the Asian bees have ways of dealing with the Asian hornets, but our bees can't survive an Asian hornet attack. So they do need a bit of help from us.
So
I
how
would
would
say.
you breed for that?
Well, we breed against aggressiveness, so we're doing the opposite thing. But there's a lot of things that bees can't adapt. They can't adapt as quickly as we're changing the environment, especially now, which each season is completely different. They're dying quite a lot, and then you can have bad winters and stuff. Yeah.
Mike is saying thank you. Any other questions?
I have a question for the compatibility locus that you were speaking about.
Yeah.
Because in the example it was biallelic. Is it actually biallelic or like highly polymorphic?
No, it's highly polymorphic. You can have up to like. It's been shown. I don't know if this is true, but it's been shown that when populations are closed, they produce new alleles if they're really strained for it. So I don't know whether this is a fact of that. Just the rarer ones are picked up, or they actually mutate the locus to sort of be able to survive. But it is usually you would have around. Well, it's been a lot of studies between 50 and 120 of alleles in one population, so that you don't run into such problems.
So is the region big?
Well, the thing is, we still don't know what kind of polymorphism it is. Is it length? Is it sequence? I think it's a mixture of everything. It's a mixture of length, locus, sequence, expression, morphism. So the final product has to be similar enough, but that final product can be affected by the length polymorphism, the sequence polymorphism, how it's expressed, how it's spliced, how it's processed. So I think it's all of that. And then if the protein that comes out is similar enough, I think.
Yeah, that's what I was wondering, because you were speaking about like a recognition rate that is much higher. So I do expect like highly polymorphic, but I would expect like a very strange protein out of this. Like, do we have a clue of what that's coding for?
There's not a lot of research on that. No, not yet. Thank you.
Okay. If no any other questions, we are done with today's presentation. Thank you, Jana.
Thank you.
Next week we have Joseph Matt talking about oyster breeding. So another interesting species that I know nothing about. So please do join. Thank you, online people.
Thank you.